Today, I'm going to talk about some of the work that we've done that led to the Pfizer BioNTech vaccine, COVID-19 vaccine, but really from the perspective of the being a scientific entrepreneur, as well as some of the basic science that was involved. But the fact of the matter is that we wouldn't have got to where we are today if I hadn't been working as a scientific entrepreneur for quite some time, as I'll try to indicate. I do have a number of conflicts of interest. I'm a founder of Precision Nanosystems and also of Acuitas Therapeutics. I'm founder and chairman of Acuitas. So just to illustrate the point that, that I am a scientific entrepreneur, I've spent most of my career working half-time in academia and half-time in companies. And so this goes right back to 1978 when I first joined the University of British Columbia. In the middle 80s, we formed a company called the Canadian Liposome Company. I was then the chief scientific officer of a company called Inex Pharmaceuticals. And then three not-for-profits that I've been involved with, the Center for Drug Research and Development, the Personalized Medicine Initiative, and the Nanomedicines Innovation Network. And this has been combined with with my academic positions as indicated below. So I've co-founded 10 startups and three not-for-profits. Makes my life kind of complicated. So I just want to go through, actually I'm going to go right back to the beginning. An alternative title here, aside from the one dealing with the BioNTech vaccine, is Why Be a Scientific Entrepreneur? And I'm going to ride that one through in terms of how the talk is organized. So why be a scientific entrepreneur? This is aimed at PIs in universities as well as postdocs and graduate students. So there's a number of compelling reasons. The first is to provide a benefit for the Canadian taxpayer. Second, if you're a professor, create jobs for your students, or if you're a student or a postdoc, create your own job. Avoid writing grants, always a fundamentally important objective. Have fun, and most basically do new and exciting science. So why be a scientific entrepreneur if you're a professor? And you could almost say it's your moral duty. For more than 40% of our PhD graduates in science, technology, engineering, and math take jobs outside Canada. And so we're really exporting a large proportion of our highly trained graduates due to the lack of jobs in Canada. And this is an older stat, 350,000 Canadians in the Bay Area, 100,000 in Boston. There's probably more than that now. So this really doesn't benefit the Canadian taxpayer who's funding our grants and the training of our students. So just think about this. If you assume an average cost of $200,000 per graduate student, and the 40% of the 5,000 STEM PhDs, if 2,000 people go south, 2,000 of these PhDs go south, we're essentially paying $400 million a year to support US industry. And I'm sure this is not something we really want. Not only that, but we're exporting wealth capitalization, wealth creation capabilities. Now, Accelera, which is a recent success story in Vancouver, has a market cap in excess of 10 billion. We'll take it as 10 billion for round numbers. They employ over 250 people now. It's 100 of those are probably PhDs. So that's wealth creation of 10 million per PhD. So if the average PhD we export only creates 1/10 of that, we're losing $2 billion a year, you can say, in wealth creation and the tax base that implies. And this certainly doesn't benefit the Canadian taxpayer who's funding our grants, et cetera. And so that's a compelling reason. Creation of jobs. If you're a professor, creating jobs, and if you're a student, creating your own job. Now, we desperately need to create jobs for our graduates, for our PhD graduates in particular. There's academic jobs for only 1 in 8 of our graduate students and postdocs, as indicated here. This is kind of an older slide, but it's certainly it remains true today. This is about the ratio of the academic positions that are available for the people that we graduate. Now, I would say that this can be done. These jobs can be created. And so this is just some of the nanomedicines and drug development companies I've co-founded in British Columbia. The basic point is, as a result of, say, GeneVant, Acuitas Therapeutics, Precision Nanosystems, Evonik Labs Canada, we have close to 300 or just over 300 people employed in these areas, and these are high quality jobs. So let's add to this some of the companies that we founded in the area of personalized medicine. So nearly 400 people in Vancouver are now participating in Vancouver being a global innovation hub for nanomedicines. 5 of the 13 approved nanomedicines have originated in Vancouver. And the Nanomedicines Innovation Network, of course, that we're using as the ready to spread these ambitions across Canada, and an aspiring global innovation hub for personalized medicine with 4 companies employing approximately 75 people. So this is just to point out it can be done. It is not what's the word, overly ambitious to think that one can do this. One can achieve these objectives by starting by startups and other means. Avoid writing grants. I'm saying this rather facetiously, but the writing grants is certainly a very frustrating exercise, and the success rate for CIHR grants is only 15% in 2020. It's remained that way for some number of years now, and it really makes grant writing to be I was going to say a futile cycle. That's not quite the case, but it is really an enormously exhausting undertaking to get grant support. And this is not true just of Canada, of course. This is a worldwide phenomenon, but it is an exercise that's not much fun. This is just indicating here that in real terms, the amount of money in the granting system has not increased significantly over the last 10 years. And so I would say that the granting system is only a very small fraction of their gross national product, 1 or 2%. And so there's 98%, 99% more money potentially available through other sources. And so I'd say writing a business plan is really much harder than writing a grant. You just have to include an economic justification for that. It requires a different mindset, but if you can write a grant, you can write a business plan. The fourth thing here is to have fun, and I want to emphasize that that's been a driving thing in my career. Is how do you do these kind of things and have an enjoyable experience in that process? So I want to tell you a little bit about the Lipex biomembrane story, starting with my research group that was started at UBC in 1978. And so we spun off Lipex biomembranes in 1985, and that subsequently was merged with another company we started called Northern Lipids, and subsequently bought, as I'll indicate, by Evonik in about 2015. And so in 1984, we invented a device for making unilamellar liposomes, and we call this device the extruder, and you'll see why when you see what the machine does. And we formed a company called Lipex Biomembranes, to market this instrument. And we knew that companies had to have a mission statement, so we developed one. And our mission statement was the best mission statement actually we've ever come up with. It was free beer every Friday and parties whenever possible. And this is certainly fun was a major objective of Lipex biomembranes. It really sprang out of the fact we developed this device, this extruder, And I gave a talk in New York, and one of the people in the audience said, "Well, I would like to buy one of those." So I put him on to the machinist we had make it for us. And then about 2 months later, I got this irate phone call saying, "Well, this thing doesn't work." And of course, the machinist who made it had no idea what it was used for, so he didn't know no quality testing or anything like that. And so that's when we realized that we had this opportunity to form this company. So it's a pretty simple instrument. You can disperse lipids in water and then ram them through a polycarbonate filter under high pressure gas. And so this instrument is indicated here. We had a couple of embellishments. We could actually put a water jacket on this thing, and so we could do whatever temperature you wanted. So we called that the thermal barrel extruder. Producing these unilamellar systems is indicated on the right-hand side. This little bar down here is 100 nanometers. This turned out to be a bestseller. There's over 3,000 of these instruments that have been sold. There's probably more than that. Certainly approaching $20 million in terms of sales now over the period from 1985 onwards. Actually, there's now $1 million extruders being sold for commercial purposes that are able to do the scale up required for clinical quantities. You can say this is free beer every Friday forever. It was certainly successful in its aim. We had a good board at this company. It was me and four postdocs in my group. It was the board of Lipex biomembranes when we started off. And of course, we thought that every serious company should support the arts. So we formed what we called the Royal Lipex Ballet, which had some notable performances, unforgettable performances. Probably we'd like to forget about some of them, but anyway, at unforgettable parties, and this is just indicating one such event. So this is just indicating how you can really start things off. And I'm posturing in a fairly lighthearted way, but it was quite serious. But start things off in a way that didn't require any investment. We didn't have any intellectual property, and yet it was hugely successful. If you have a good idea and you have something that's it's a reputational quality to it, things can work out pretty well. As I mentioned, we sold Northern Lipids to Evonik Industries. It's a very large company of 36,000 employees. In 2016, they're now expanding the facility in Vancouver with over 100 employees, and it's expanding rapidly from there. Okay, so have fun. The fifth area is do new and exciting science. And so the second half of the talk, I'm just going to focus on that. So this concerns mainly now the Acuitas Therapeutics story. So starting from my group in 1978, we started a company called the Canadian Liposome Company, which was a subsidiary of a US company based in Princeton in New Jersey. That morphed into Inex Pharmaceuticals, and from there into Acuitas. And the first Canadian liposome company and Inex, to a large extent, were focused on anti-cancer drugs, whereas Acuitas is focused on gene therapy applications. So new and exciting science. Our initial focus in the early 80s was, how can we deliver cancer drugs more effectively? I keep on saying this, that we have a completely ridiculous situation at the moment, where a patient with disease, cancer in various locations, we inject them with a drug that goes everywhere in their body. Only 0.001% actually gets to a tumor. It's a miracle these drugs work at all. We really do need delivery systems that enhance delivery to disease sites and and protect sensitive tissues. I mean, this is, I think 50 years from now, people are going to look back and you say, you actually use medicines that went everywhere in your body and not just to where they were needed. This is a transition we really have to make. And so this is where some of the science comes in. How do we load these cancer drugs into liposomal systems? And we've been doing it for basic science applications for looking at, the effects of ion gradients in biological membranes. We've been measuring the pH gradients across those membranes using methylamine, which is a weak base. So as a weak base, methylamine has this property. It can exist in a protonated or charged form, and it can exist in a neutral form when it gets deprotonated. Now, the neutral form is completely permeable through lipid bilayers. And so if you have a pH gradient across a membrane, say pH 7 on the outside and pH 4 on the inside, then the neutral form can go in. But once it gets in, it gets protonated, and so it then becomes charged, and it can't get back out again. And so at equilibrium, you can do the mathematics here, the concentration on the inside over that on the outside mirrors the proton gradient. So if you have a 3-unit pH gradient, you have a 3,000 times higher concentration of the methylamine on the inside than you do on the outside. And so this was, kind of an academic interest and observation. And so one day we realized that a lot of anti-cancer drugs were weak bases. So this is doxorubicin indicated on the left-hand side. And we found that if we had these liposomal systems with a pH gradient, we could load them up to such a high extent that the drug would start to crystallize out on the inside of these liposomal systems. And so we can get huge numbers, hundreds of thousands of molecules of the drug associated with these 100 nanometer diameter systems. So a highly efficient and really very straightforward way of loading them up. So this attracted the attention of a company called the Liposome Company in Princeton, New Jersey. They funded the Canadian Liposome Company. And we had 2 drugs that resulted from this, an amphotericin or antifungal drug and a anti-cancer drug that encapsulated doxorubicin. They were approved in the middle 90s and in 2000. So the Liposome Company abandoned us in 1991. They got a new CEO, couldn't understand why he had this company up in Vancouver. So we had to regroup, but we knew what to do. So we formed our own company, Inex Pharmaceuticals, in 1992, again, focused around anti-cancer drugs. But the point I want to make now is that as a result of starting our own company and getting going in this cancer field, you end up doing things which maybe you wouldn't have done in an academic environment. This just illustrates the situation that we ran into in the middle 90s. The CEO that we'd hired to run the company, Jim Miller, said, I can't raise money putting old drugs into liposomes. We need to be doing gene therapy, because that was, at that time, as it is now actually, had become really a very hot topic in that period. And so we said, okay, gee, this is kind of a challenge. We'd been working and were quite successful loading drugs like doxorubicin into these nanoparticles. How could we possibly load these macromolecules? SRNA is pictured here. They're 13 kilodaltons in size. And so we immediately hit a problem that in order to load these things into lipid-based systems, we had to use positively charged lipids. Now, there's no cationic lipids in nature. They're really very highly toxic. There's net neutral lipids or negatively charged lipids. And that's really because they tend to rip membranes apart. They combine with the negatively charged lipids in membranes and destroy the bilayer structure. So to reduce this toxicity, we decided to try what we term ionizable cationic lipids that have this property that is, say, neutral pH. They're net neutral, but at low pH, say pH 4, of the acidity of a lemon, they get protonated and are positively charged. And so this was our early version called DODAP that has this property. Now you might ask, where did we get this idea from? Really got it from basic science. We'd been in the lab, we'd been trying to study lipid asymmetry that we could induce in response to pH gradients. And we could flip this molecule across the membrane, say if we have an acid on the inside, and go across the neutral form, be protonated and stay on the inside. And so we could see all sorts of morphological consequences of that. However, the problem came is how we encapsulate these polynucleotides. Then this was a logical molecule to investigate, and it turned out it worked, and it worked very well. So we developed a rapid mixing process, dissolving the lipid in ethanol, mixing with the oligo in pH 4, and developing these systems. And you can see intuitively how it is. You can get high encapsulation efficiencies. And we could regulate the size according to the amount of surface lipid, the ratio of the surface lipid to the core lipid. A surface lipid in this case is a PEG polymer. So we invented here a new class of lipid nanoparticles with a hydrophobic core as opposed to an aqueous core, ideally suited to encapsulation of negatively charged macromolecules such as RNA and DNA. Effective encapsulation efficiency is approaching 100%, stable, monodispersed, relatively non-toxic, scalable, and reproducible, and so on. And so after that, I was attending a conference, or just during that time, I was attending a conference in London in 2004. And Victor Kateliansky, who was then the VP research for NMIN Pharmaceuticals, really accosted me. He pursued me through Soho, as I remember, saying, we have a delivery problem. How do we get our small interfering RNA, pictured here, into hepatocytes in vivo? And so we set up a three-way collaboration between my laboratory, Inex, and Alnylam to try and conquer this problem. The result of this was really the most successful collaboration I've ever been part of. It went on for about 7 years. We tested literally hundreds of different lipids, constituents, and tested all of those in animals. And it resulted in a formulation that was highly effective for silencing genes in the liver. And so this is indicated here. We went from about 10 mg per kg in terms of potency down to about 5 micrograms per kilogram, and ended up with a formulation that we could then use to treat a disease. And so this was pursued by Alnylam to treat a disease called transthyretin-induced amyloidosis, which is caused by production of a mutant transthyretin in the liver, results in formation of these fibrils in the circulation, which cause cardiotoxicity and neurotoxicity, fatal within 5 years. And so our hypothesis was if we had an SRNA to silence the TTR gene in the liver, then this would stop the production, reduce the levels of fibrils in the blood, and then perhaps even reverse accumulated damage subsequent to reducing the amount that's in the circulation. So this resulted in a clinical trial that started in 2012. The results were announced in 2017. It hit on the primary endpoint and all secondary endpoints with extraordinarily high confidence values. We'd taken really a delivery system to liver, a small interfering RNA to the liver, and we have now an essentially curative, or at least it can contain the disease for a previously fatal disorder. It was approved by the FDA in 2018 and is now in general use and very successful in that. So we then formed Acuitas Therapeutics to say, okay, well, let's take this a little further. Can we extend this kind of technology from siRNA to much larger messenger RNA molecules? And so this is the situation where we started off with this. We started off, first of all, of course, with doxorubicin, which is a good deal smaller than this. But I tried to do this to scale with the mRNA for say luciferase. But if I did it really to scale, you wouldn't be able to see this tiny dot here for signifying siRNA. Point being that we're trying to encapsulate a much larger molecule. But we found the same process could be used for encapsulating mRNA. Also, when injected intravenously, we get expression of the protein that the mRNA is coding for in the liver. And then we went through an optimization process, at least Acuitas did, to try to get maximum protein expression. This is where it pays really to get out there and be doing something that could have, you're going in one direction. We were trying to produce proteins that would be of therapeutic use in the liver. But then we were approached by Drew Weissman from the University of Pennsylvania, who said, we have a delivery problem. He's an immunologist. He was wanting to use mRNA as a vaccine. How do we get mRNA coding for viral proteins into muscle and immune cells in vivo? And he'd worked for many years with Catalin Carrico on these vaccines, and they had developed mRNA that once it gets inside a cell, had very high levels of gene expression. And so what Drew and Catalin, and Drew in particular, wanted was this delivery system be packaged into a optimized lipid nanoparticle, which could then be injected intramuscularly to produce MHC1, MHC2 class immune responses, and thereby prime the immune system so that subsequent infections that would be avoided. So the mRNA here, of course, is the spike protein of the SARS-CoV-2 virus. So to cut a long story short, the Pfizer and BioNTech then took that forward in a phase 3 study of COVID-19 vaccine, which met all primary efficacy endpoints. This results announced in middle of November in 2020, 95% effective, beginning 28 days after the first dose. The efficacy consistent. This is being borne out now obviously in major studies as this vaccine is finding its way into the general population. Older people, et cetera, over 94% protection, relatively safe and well tolerated. The companies are now expecting to produce up to 2 billion doses by the end of 2021. I think you're going to see it go up from there. So these systems are playing a major role in ending the COVID-19 pandemic. So what I want to say is that, this is by virtue of starting out in one area and pushing forward, you do have this opportunity to take advantage of unexpected opportunities, and certainly the vaccine application was an unexpected opportunity, but it's having major ramifications that we're all aware of. The vaccine applications are really just the tip of the iceberg. We're going to see this lipid nanoparticle mRNA technology enabling a multitude of gene therapies. Vaccines for HIV, Zika, influenza are certainly coming into focus in very direct ways. Chronic diseases, inherited diseases, they're all potentially attackable using this approach. So I hope I pointed out to you why being a scientific entrepreneur is an important thing to do. Provide a benefit for the Canadian taxpayer, create jobs, avoid writing grants, have fun, and most importantly, well, I think having fun is probably pretty important too, but do really new and exciting science, and it does drive you in that direction, and actually make those academic discoveries into products that really help people. So I'll close there. I think I've made my point that as a result of being, this would not have happened if I had stayed in a university environment and not ventured out into the commercial world. This, of course, also implicates a huge number of people, hundreds, probably thousands of people that I have to acknowledge, both in Acuitas, Precision Nanosystems, Arbutus, Alnylam, my own lab, and Drew Weissman, University of Pennsylvania, the breadth of interactions that come out as a result of venturing into these startup companies is enormous, and it's extraordinarily rich experience. So I would make one other point, and that is that this really results from, as I tried to emphasize, people that I've collaborated with for over 40 years, and which is a remarkable thing in itself. Big hope Tom Madden, Marcel Bally, who's not on here, but it's as a result of that long-term collaboration, which we could only do by spinning out companies that could keep people employed and focused, that we've had the success that we've had. Okay, so I'll take any questions that people may have. Thank you.
So Devika states, great talk, thank you. In your opinion, what would be some of the LNP characteristics that should be optimized for targeting LNP delivery to non-liver targets?
Yes, well, from an IV intravenous administration point of view right now, we're very good at getting to the liver, but not necessarily to other tissues. And so there's two stages to targeting. One is actually getting to those other tissues, and then maybe targeting particular cells or tissues in that environment. And so the next step that we need are in terms of an IV administration, are systems that are long circulating. In other words, instead of a 15-minute circulation lifetime, they might have a 10-hour circulation lifetime, and therefore be much more accessible to other tissues in the body. Subsequent to that, once we achieve that, and we're nowhere near at the moment, having systems that are transfection competent, but also circulating for a long time, then we want to find ways in which we can make them, approach one cell population versus another. This is going to be quite a long journey, but that's the way that I see it progressing.
The two questions are, what is the closest thing to Y Combinator in Canada? And the second question is, what is the closest thing to DARPA in Canada?
Well, I'm not too familiar with the Y Combinator, so I'll go on to DARPA. There is within Department of Defense, there is a research unit which is concerned with, threats to Canadians. I forget the name of the organization, but they do fund basically investigations into things that would protect soldiers in the battlefield, or potential terrorist, bioterrorist ops, things that incidents that may be potentially possible. The DARPA certainly has a much larger budget, and it has a formidable reputation. But we do have equivalents here in Canada.
Okay. The next question is from David Holloway. Marshall, could you read that question out?
Yep, of course. David Holloway asks, could you talk a bit more about how to target the liposome to particular organs, for example, the liver?
Yeah, from the liver point of view, we're taking advantage of a you might say a natural targeting process. It turns out that these systems that we developed have optimized systems adsorb apolipoprotein E once they get to the liver or close to it, and the ApoE is then facilitates uptake into hepatocytes via the LDL, low density lipoprotein receptor, and also some scravaging receptors. So that's the targeting process that's really being employed there, and we discovered that it was a serendipitous observation as a result of working in vivo to try and optimize the systems. They're treated like chylomicron remnants. Chylomicrons, take fats and so on from our diet, distribute them through peripheral tissues, and then the remnants come back and adsorb ApoE and then are taken up into hepatocytes. And it turns out that we're kind of hijacking that process. Whether we can use that same process to target, or related processes to target other tissues, is something that I think is going to be a fertile area for study in the future. That the protein corona that from the serum proteins that adsorb to these lipid nanoparticle systems very much affect the particular cells that they will be taken up into. And so I think we're going to find some real advances there. That can be the protein corona can be manipulated by changing the lipid composition and other factors associated with the LNP. So there's an interesting, I think it's a fascinating area of study in terms of getting different selectivity in different tissues.
Thank you. Marshall, the next question I saw pop up was from Anna Alexieva. If you could read that question out, please.
Sure. Her question is, what are the differences in siRNA and mRNA formulation preparation?
Well, first of all, for the mRNA versus the siRNA lipid nanoparticles, there's different ionizable cationic lipids that have been found to be optimized for one application versus another. I should say that that's not completely established yet. That the ionizable lipids that are used in mRNA, optimized mRNA systems probably work pretty well with siRNA as well, but I haven't seen work that might demonstrate that. But otherwise, the systems are pretty much the same, except for one important factor, and that is that of course there's many more copies of the of say siRNA in a lipid nanoparticle than there are of mRNA. And you can do the calculation, but say in a non-patro formulation, there's roughly 700 copies of the siRNA per 80 nanometer, 60 or 70 nanometer lipid nanoparticle. For the mRNA, you might have as little as 2 or 3 copies, depending upon the size of the mRNA. So the actual carrier is different simply by virtue of the number of copies of the active ingredients that are associated with each of the nanoparticles.
Okay, thank you. So there's some questions in the chat. The first one I've got here is from Nancy Ford. She says, I'm curious about the timeline of getting mRNA into immune cells. When did you start to tackle this problem with Drew Weissman and Kathleen Carrico? When did you succeed? When was mRNA for the spike protein first put into LNPs?
These are some questions I really don't know. I don't know the precise chronology, but as I was indicating, and at least I think I indicated in the talk, the focus of Acuitas in say 2012, 2013, 14 was primarily on getting optimized systems that would produce proteins in the liver following an intravenous injection. Now, Drew Weissman came on the scene somewhere in that time frame. I think it was 2014, 2015, saying I would like to test these systems as vaccines. The interaction with BioNTech happened subsequently, but I think that was mainly focused on cancer vaccines. And so it was only in January, February of last year that the switch to the SARS-CoV-2 application from a vaccine point of view was made. I mean, this is done with startling rapidity when you think about it, because things went into clinical trials. What was it, April, May kind of time frame, from a standing start in say January, February sort of time frame. But that's a rough chronology. I'm sure other people could give you a more precise picture.
Thank you. The next question is from Tom Liu, who asks, what is the difference of delivery system between Pfizer B and Moderna COVID vaccines?
The difference, well, there's a number of differences. I can't really comment on the Moderna one. I do know that the ionizable lipid is different. And of course, their mRNA is different as well. So those are the primary differences as far as I'm aware.
Okay, thank you. Another question from an anonymous person on the attending. What are the main obstacles for establishing LNPs for drug delivery in cancer treatment?
Oh, the main obstacles. I guess the main obstacle is getting a really robust immune response to the epitope that you want to get an immune response to. And so again, this is a matter of potency. Now, there are some of these formulations going in or in clinical trials now. But I'm not really up to date with how people are doing in terms of the robustness of the responses. The idea here is quite simple, of course, because you're just you're saying, okay, let's do a biopsy, find out what are the proteins that are peculiar or just individualized to the tumor, make an mRNA that codes for those epitopes, and then raise an immune response to it. Now, there's a number of issues. You don't want to raise an autoimmune response to proteins that are already present in the body, et cetera, et cetera. And there's a potential for using a multi-mRNA approach where you're going after many targets on the tumor. I would say it's fairly early days for the cancer vaccines, but the results are looking pretty promising that I'm aware of.
Anthony Tam states, LNPs have successes in silencing certain genes produced by the liver, and then asks, do you see a potential from an investor's perspective for LNPs / siRNA targeting some to all hepatitis viruses as an alternative therapeutic treatment to existing therapies?
Yes, yeah, it's for the liver, one does have the competing technology of, say, GalNAc, targeting of the individual oligonucleotides themselves, and that's also quite effective. But certainly the the LNP approach delivering to hepatocytes is effective as well. The advantage of the GalNAc approach is you can go in subcutaneously versus intravenously for the lipid nanoparticle approach. So it's this is the approach now that Alnylam's taking, for example, is the GalNAc approach. But if you want to access other tissues, you're probably back to the lipid nanoparticle approach.
And thank you, Peter, for a fantastic, inspiring talk. My question is about advice you may have to make the process of being a scientist entrepreneur easier. So, I note that, as soon as you became full professor, you went to a timeshare of your commercial activities and your academic activities to make it possible. You also founded these 3 nonprofit organizations to make the process easier to take breakthrough invention out into making the kind of impact you've made. Any advice that you have for innovation policymakers or for university leadership on enablers or barriers or things that they could to do to make this process easier?
Yeah, there are a number of things. I mean, one of the issues in academia is you don't necessarily get many points for these these sorts of activities, say starting companies, etc. And I do think that there has to be a better way of acknowledging that within the academic system, of saying this is a valuable thing to do. Of course, it's difficult for this is kind of for universities to this whole interaction with industry is fraught with a number of issues, conflicts of interest issues, and others. And so having a very clear process that that PIs can follow is also essential here. But I think the basic thing is to say, look, this is really quite important to do. You know, there's a certain level of things we can do in universities, but when we get into actually making a drug or whatever to treat a disease, you cannot do that in a university. And I realized that really early on in my career. You know, if you want to take a drug through clinical trials, it's $100 million. Well, it's just not on to imagine that the academic system can provide that sort of those sort of resources. So you do have to get out there in a commercial environment. And I've had other experiences where if you try and license, you know, something you've developed in the lab into a receptacle company, that this relies very much on a champion in that company, you know, pushing it all the way through. And it's hard, that's very, in all three cases that I've done, it has resulted in the drug basically not being developed subsequent to the licensing process. You need somebody that's really passionate about the success of a particular therapy and really wanting to make that happen. And very often that's the PI or a postdoc or whatever in academia who has the idea and will do anything to push it through. And so I think this needs to be acknowledged more as an important activity within the university setting, and so that is a crucial. I think people are becoming aware that it is an important thing to do, but we haven't really got it to the point now where it's as straightforward as it needs to be.
Mm-hmm. Thank you. On a related question, Michel Meunier asks, or he says, I understand that know-how is important in starting companies, but how important is IP or a scientific entrepreneur in starting a company?
Well, it's certainly, you know, if you want to develop a drug, IP is crucial. Getting intellectual property that protects a potential investor from somebody else just using the same approach is really important, because otherwise you're not going to get the investment. And so this is again where universities help, and certainly UBC helps a lot in terms of you know, helping the inventor get a patent or patent application, and then subsequently helping them to license that intellectual property to their startup company. But certainly intellectual property is a crucial part of the picture for getting a startup going and really being able to attract investment.
Natalia Onashenko also thanks you for your talk and asks, are SARS-CoV-2 LNP-based vaccines manufactured using microfluidics?
Are they manufactured using, not to my knowledge. I think they're manufactured using either T-tube or Y-tube approaches, so I don't think that the microfluidic mixing approach is yet being used for these vaccines.
The next question, Liza Silverman asks, I was wondering why mRNA and siRNA are better encapsulated in a hydrophobic core than an aqueous core when they are charged molecules.
Yeah, it's a logical question. The basic problem is encapsulation efficiency. As you say, these highly negatively charged molecules are highly water soluble, and so if you can envision if you formed say liposomes, bilayer systems in the presence of these molecules, you'd get some of that material being encapsulated. The problem is the trapped volume of these small systems, say 60, 70 nanometer, is really quite small, and so it would end up that you would only be able to encapsulate a few percent, 3 or 4% of the total amount of sRNA or mRNA that was present. In essence, you'd be wasting all the rest of it. And so in order to increase the encapsulation efficiency, this is where you have to move to these positively charged lipids, which engender 90 to 100% trapping efficiencies.
There's a question from Ali Deshari, who asks, what is the zeta potential of the LNP system used for mRNA vaccine delivery?
It's close to zero at at physiological pH values. This is where the ionizable lipid is in a net neutral form, and of course the other lipid constituents are uncharged. What it is, the zeta potential will increase in a positive fashion as these things are formed at pH 4, but then the pH is raised to pH, to 7.4 physiological pH subsequent to that. At lower pH, where the ionizable lipid is charged, there will be a larger surface potential associated with these systems, but it's relatively neutral at neutral pH.
We have a short question from William Wang. Is it possible to deliver DNA successfully?
Oh yes, yeah, no, it's possible to deliver it in the sense, in the same way you can deliver mRNA to the cytosol, where the cytoplasm, where the where the ribosome, where basically the mRNA can get translated. For DNA, you have this additional problem that it has to get across the nuclear membrane, and we haven't yet really found a way of doing that. In terms of transfecting using DNA, you really have to rely on the targeting really rapidly dividing cells, and then as cells are dividing, then access to the nucleus is dramatically enhanced, and you do see transfection in those cases. But for other cells, that's not the case.
So the next question is that LNPs have been successful in penetrating various tissues. I was wondering, however, after penetrating the tissues, is there significant differences in how effective the payload, either mRNA or siRNA, behaves in different tissues?
Yeah, I don't really know. It's obvious. I mean, the two major routes that have been explored at this point are obviously intravenous, and there, a very large proportion of the material gets to the liver and is able to transfect, hepatocytes, etc. The tissues that we're transfecting when we go in intramuscularly for a vaccine application, clearly we are transfecting some tissues there. It's not quite as clear what cells exactly are the dominant feature, but are the dominant cells that we're transfecting. In terms of a journey through extravascular tissue and whether that affects the payload, at this point I would say it doesn't seem to affect the payload. However, on that journey, whether or not if there's one cell type versus another that's more readily transfected, the answer is not really in yet on that.
Okay, thank you. Francisco Pesene has a question. What can you tell us about the thermal stability of the carriers with SI or mRNA?
The thermal stability of carriers, the carriers with sRNA are really quite stable. There we have in an aqueous form approximately, a year of stability as far as I'm aware. Now in the case of mRNA, that's not the case. The mRNA is a more, those systems are definitely more sensitive to breakdown. I have a feeling these are problems that are going to be beaten over the next while. But right now, that's where we're at, where they have to be stored at, in some cases, excessively low temperatures.
Stavrula Zagku thanks you for your inspiring talk and says, I'm a PhD student working on PLA particles. I was wondering, what is the next big thing in nanomedicine, and if there is a way to facilitate delivery in the brain?
Well, certainly having systems that work in the brain would be a next big thing. it's unlikely, in my view, that that's going to happen by getting things across the blood-brain barrier following an intravenous injection, for example. And so I think what you're going to see is, is a stepwise approach. We're probably going directly into the brain via intrathecal or other means. And so that's the, these are things that, as I keep on telling people, at the moment we're in a bit of a gold rush of opportunity, and for basically accessing different tissues with different cargoes for different disease opportunities. You certainly are going to see systems coming into play that are that will be useful for intraventricular, intrathecal, direct injection into the eye. We're getting close to having systems that are going to get to bone marrow. The vaccines are going to be, become more and more potent. That you can see ways forward in all of these areas, and so there's a number of next big things. I guess you can sort of take your pick here in terms of opportunities, huge opportunities that are coming up. I'll just leave the answer there. It's open field running at the moment.
Well, Dr. Cullis, it's my pleasure to thank you on behalf of all of the attendees for your lifelong commitment to excellence in science, which has led to one of the vaccines that will help to ameliorate the non-travel that we're experiencing as a result of the COVID-19 epidemic. Thank you also for launching the NMIN Lecture Series with such an inspiring talk today. And to our participants, thank you for joining us for this inaugural NMIN Lecture.